and we're going to dive right in today to a new concept to many of you called redox dysbiosis or oxidative stress, which is an environmental environment that happens within our colon that we're starting to appreciate when it's in the wrong place at the wrong time, similar to how we used to think about gut microbiome, bugs being in the wrong place in the wrong time in the context of SIBO. Turns out the wrong oxidative stress environment can lead to all these downstream things that we think about when we think about IBS and SIBO in particular, but lots of other chronic diseases as well. What do we mean by redox? When we'll get into some of the basic science, but the environment of sharing electrons is sort of at the heart of this. And microbes in our human cells are just making energy. And how we make energy fundamentally is through a process of electron exchange. And when that balance is out of the tipping point of where it should be, we think about it as oxidative stress. We've heard about things like oxidative reactive oxygen species, or when you think about grilled food, you sometimes are thinking about advanced glycolation end products that cause oxidative stress. Those are just examples of things that we've heard about in food, but now we're going to put it in the context of the whole microbiome and what we're seeing with this as an organ system and how it gets out of balance, you end up with disease. And the reason this is a new conversation is in part not because it hasn't been here the whole time, but because we haven't really been able to see it or observe it. The gut microbiome at its core is fundamentally an undiscovered organ system until just several decades ago, when we were able to use genetic sequencing of the microbes to get a picture of what this environment looked like. And since then, in those last 20 years, and in particular in the last 5, we've really started to appreciate that these little microbes make a huge, diverse array of metabolites or postbiotics, which again we'll define later, but the small molecules that this microbiome makes and communicates with us, its metabolic output is central to how our body functions. And it's a dynamic system, it's changing. There's lots of things that are really cool about it that are different than other components of our organ systems because it's microbes and they're changing and evolving so much faster than us. And today, I think the goal is to dive right past the basics of the microbiome, appreciate that it's an organ system, but then help you understand what happens when this redox environment gets out of balance and how we can start to use that as a frame to make decisions about health and our journey towards getting back into balance. The microbiome has been thought of as a pantry or the reservoir of nutrients that we cannot make in our human cells. We think about diet and essential amino acids and vitamins as things we get from food, but in addition to that, the microbiome is making a lot of these nutrients and vitamins that we didn't used to think about as coming from the microbes themselves. I have been thinking a lot about the microbiome and its state in the current Western world and how we've depleted a lot of its diversity. And the same way we think of scurvy from the olden days with pirates and not having enough vitamin C, we essentially are looking at scurvy of the modern age with this microbiome inability to make these core nutrients that are our reservoir. Patrick, do you have anything to add to this concept? I know it's something you're excited about too.
I think it's like when we begin to look at quorum sensing and the interrelationship of all the organisms, that they work together as a superorganism. And the Pantriome is taking that quorum sensing idea and expanding it to be able to say that it has to do with all nutrients, which functionally means that the taxa that we talk about are not nearly as important as the functionality of what's happening within the microbiome itself.
Yeah. And, we are just, again, starting to discover this organ system, but there's some basic principles that we're now revisiting that we've known about on the human cell side that we can now start to apply to these microbes. So, how do the microbes make these nutrients or these things for the pantry? Well, we go back to the central dogma of biology, which is how do our genes get turned into proteins. And just the same way that our cells have genetic information, DNA, that gets converted into RNA and then expressed into proteins, the microbes are doing the same thing in our gut microbiome in particular. And the proteins that the microbes make are often a type of protein called enzyme. And an enzyme is important because it can then take a precursor molecule like the broccoli you're eating or inulin and convert it into an end molecule like butyrate, which is something we talk a lot about in the context of the human microbiome. And so we'll revisit this later, but I just wanted to bring us back to some basic biology that these microbes and the way that their genes are expressed impact this pantry's reservoir of nutrients, in addition to what those precursors are and the environment it is. So, the microbiome and its genes are the building blocks of this organ system and our reservoir of nutrients that interface with our immune system, our liver, our metabolic, our cardiovascular, our brain, you name it. And the way that it's interfacing is these small molecules, metabolites, or what I'll call postbiotics, are entering into our system and interfacing with receptors, both in the gut, but also throughout the body. And what we're starting to appreciate, which I don't think was common knowledge even a decade ago, is that the microbes, metabolites that they're making, these nutrients they're creating, are traveling to all the different parts of our body, including our brain and our muscle tissue and our bone marrow, and impacting cells throughout the system, not just locally within the gut. So, our host or human cells that line the gut are called colonocytes. And they're really important for modulating this environment, in particular, the redox weather, if you will. And butyrate is a postbiotic or metabolite that a healthy microbiome that has a good mixture of microbes consuming fiber and converting them makes. So this is an example of this process where having the right bugs, we think of things like Akkermansia or fecal prosnitziae, convert the food you're eating into what we call postbiotic or a butyrate as an example. And then these feed those cells, those colonocytes that line the gut, and provide an oxygen sink and balance this redox electron transport, and then allow the colonocytes to make energy and feedback loop into creating this environment. We'll get into some of the details and revisit this some more, but I just wanted to provide an example of how do the microbes in your gut creating a metabolite interface with this whole system. And butyrate's one of our most studied examples because it can be this energy source for the colonocytes. It can bind immune cells and initiate anti-inflammatory patterns. It can help with our leaky gut and keeping those colonocytes stuck together and happy and not letting things leak through. And it can even modulate the redox environment directly as an antioxidant or through binding our cells and expressing things that are antioxidant.